Hi, and thanks for tuning in. So today, we're not going to learn anything new, but instead, what I'm going to do is show you a neat trick that I show my students about how to remember all the things that are going on in the immune system. So this is what I call the immune system drawing, and it's a neat way to help take all of the information that you've learned and boil it down into something that's easy to sort of learn from. I've found that students most often are more successful in learning complex topics when they can put it in some type of visual format. And the cool part about this drawing is I'm going to encourage you to do the drawing on your own. Don't just watch me do the drawing, because if you continue to try to perfect this drawing and repeat doing this process a number of times, it's a form of active learning where you'll begin to learn the information better, but also it will help to highlight the parts of the immune system that you may be struggling with. So when you get to a point while you're doing the drawing and you can't remember what's next or why you drew something that you drew, stop and go back and look at some of the videos and try to remember. Look at some of the notes you may have taken or whatever textbook you have, and go, oh, by the way, why did he draw this, or what's the next step in the process? Don't just rely on trying to memorize my drawing. The other cool thing is it's going to become your drawing. You can add things or you can remove things as you see fit. But I found students have used this quite successfully in the past as a way of trying to remember what's happening with the immune system and put some pictures and some drawings to what's actually happening. Now, the one thing I want to stress to you is I'm going to draw it one way. There are lots of different ways to draw it. And remember, when we talked about the immune system, there are lots of ways the immune system can be activated, and not everything always has to happen in the exact same order. So just realize this is a sort of one interpretation of what can happen. You may have a different interpretation. You may want to do one specifically for what happens if it's a bacterial infection compared to a viral infection, for example. So I'll go ahead and get started at this point. And like I said, you can make this your own. So here's how I'm going to start. I'm going to start by talking about some type of bacterium that's made it into the tissue. So you may notice that I'm drawing it red and it's sort of rod shaped. So I'm going to go ahead and assume that whatever's invading my patient here is a gram-negative rod, and I drew some flagella on the back end so that it's flagellated. It could be something like an E. coli. Now, obviously, this thing has made it into the tissue, so we've already passed the first line of defense. Remember, the first line of defense really isn't worth drawing all that much because it's not an active response. It's just what's preventing things from getting in. And obviously, if we're talking about an immune response, we've got to start pretty much with the second line of defense. So one of the first things that any pathogen is likely to encounter if it makes it into the tissue is that really hyperactive protein defense system called complement. So, and we can draw some complement maybe that's now starting to bind to the surface because it's quite promiscuous and it'll bind to anything foreign that's made it in your body. So let's talk a little bit about what complement is doing in this process. So remember that complement has many uses. It's an incredibly useful protein system for our body. So the first thing, just the sheer act of complement binding to the surface of something sort of labels it as foreign and makes it easier to eat, right? So the first thing is that opsonization. So opsonization is just the labeling of anything, the binding of complement to the surface. We also talk about the fact that antibodies can opsonize as well. So opsonization actually has a couple of functions. If you remember that it does makes it easier for your phagocytes to eat them because they have those complement receptors on their surface to help bind to and eat that. The other thing, it's going to be important later on for B cells, right? So B cell activation. Remember, we need the follicular dendritic cells also have those complement receptors that can bind to that complement opsonized antigen to activate B cells. The second thing that's gonna do for us is it's going to cause. It kind of acts as a chemokine. So remember, complement is activated by breaking a piece of it off, right? So you cleave complement little pieces. So the active parts will bind to the bacterium or whatever the pathogen is, and these other little parts, they run around in the tissue. They act as a chemokine. They can act as a chemoattractant cytokine to bring other organisms or other cells to help fight this off. So you can draw little pieces of chemokine complement starting to secrete from the area. And then the third thing it's going to do is it can form those membrane attack complexes. So remember, MACs. So as they begin to recruit and initiate the remainder of this pathway, you're going to start forming these membrane attack complexes on the surface, and that can actually punch holes and destroy that particular bacterium, which could kill it. Now, of course, if we ended this conversation just with this bacterium and all of its buddies, because it's probably not going to be alone, there's going to be other bacteria there with it, right? If complement ended this whole invasion, as it were, then we would stop the conversation and there'd be no point in having this talk. So we're just going to assume that's not what happened. So there are other cells that are in our body that are actually able to assist with this particular process, and they might actually be recruited by these chemokines, for example. So let's talk first about the two sentinel cells that we have in our tissue. One of them is the macrophage. So the macrophage is that big cell. It's often abbreviated with M with a circle through a slash through it. The macrophage can make it into, as already in the tissue, and remember they are coated with these surface receptors called PRRs, which are pathogen recognition receptors. And pathogen recognition receptors are able to bind to small molecules, proteins, lipids, carbohydrates that are sort of being secreted or falling off of or being broken off of bacteria and foreign substances as they survive in our body. Remember, these little foreign molecules are known as PAMPs, right? Pathogen associated molecular patterns. So you've got all these things floating around. They also have chemokine receptors on their surface. And bottom line is our macrophage is supposed to have blue eyes because he's blue. Our macrophage is generally happy at this point in his life. He's that resting macrophage that's going around just sort of patrolling the tissue, hoping to clean some stuff up and make it so that your tissue isn't packed full of dead and dying cells and other cellular debris. So that's the resting macrophage. But when he encounters some of these chemokines that might make it out to him, that can cause his status to change, and he will go from being sort of this happy-go-lucky garbage collecting macrophage to an unhappy activated macrophage, right? Because now he's sensing that there is some danger signal coming from somewhere in the tissue. And at this point, the macrophage is going to kind of change. He will become a good antigen-presenting cell, which will be needed later on. But also, he's going to undergo a process called chemotaxis, which means he's going to move in response to those chemical cues that are in the form of these complement chemokines. There also could, at this point, in some circumstances, be some other chemokines secreted by other cells in the tissue, but not in this particular instance. See what I mean? You can kind of tell the story however you'd like. And the other big thing that's going to happen is when that macrophage makes it into the tissue near the location, these PRRs are going to start coming in contact with those PAMPs. And at this point, he's going to go from being an activated macrophage to some sort of snarling, evil, angry, very angry, hyperactivated macrophage and turn into sort of an eating machine, because now he's recognized that not only is there the body, those intrinsic signals that there's a problem, there's also an exogenous signal that this is confirmation that there is an actual problem in the tissue. So that macrophage is going to go to work. One of the things it will do once it's hyperactivated, it's going to start secreting its own cytokines in the area. And the reason why is it's going to want to start doing a couple of things. First off, those cytokines, either the cytokines that are being released by the macrophage or those chemokines in the form of complement, are also going to recruit the other sentinel cell that's in the tissue, and that is the dendritic cell. And of course, that dendritic cell, under its resting state, is also covered in PRRs. So it's coated in those pathogen recognition receptors all over its surface, because remember, as a sentinel cell, the job of this dendritic cell is to sort of taste test the environment and look for what's around, looking for PAMPs or other indications that there might be a problem. And again, whether the dendritic cell receives a direct signal of invasion by binding to PAMPs, or whether it binds to some of these chemokines in the case of those chemokines being secreted by the macrophage or by the presence of complement, this dendritic cell is going to completely change its it's supposed to be blue, completely change its attitude and it will now become an activated macrophage. So I tend to draw the things that are activated as unhappy. They're not unhappy. It's their job, but they've gone from being, hey, everything's okay, to everything's now a problem. And again, it's going to follow that chemical trail via the chemotaxis mechanism, and it's also going to come to the tissue. Now remember, dendritic cells and macrophages are both considered phagocytes, but what they phagocytose and for what reason is different. The macrophage is going to begin phagocytosing these whole bacteria and eating them, whereas the dendritic cell is more interested in the PAMPs. It's interested in collecting that battlefield antigen. Nevertheless, that newly activated dendritic cell can also begin secreting cytokines as well, and this will also recruit other dendritic cells to the scene. You will also get other macrophages coming to the scene. So basically, we're starting to recruit a bunch of newly activated immune cells that are important. The other big thing that's going to happen is that these cytokines are going to cause a change in the nearby blood vessels just outside the tissue. So remember, these blood vessels actually have receptors on their surface that allow them to interact with these inflammatory cytokines that are being released by the macrophages and by the dendritic cells. And when they bind these, they're going to begin producing new proteins, and they're also going to begin the process of vasodilation. So these blood vessels are actually going to become stretchy. They're going to become leaky, because in the end, what we're going to want is we're going to want to start bringing in blood products. We want the cells like the neutrophils. We want the natural killer cells eventually, perhaps. And eventually, we're going to end up with T cells and antibodies and all kinds of other stuff that'll be entering in from the tissue. So remember, under normal conditions, these neutrophils and these natural killer cells are kind of zipping along through the bloodstream. Natural killer cells quite often live in the spleen and the bone marrow as well. But your bloodstream is filled with something like 20 billion different neutrophils that are just sort of zipping along. And what's going to happen in order to get them to come into the tissue is we're going to have some of these new proteins begin to be produced. So we're going to be binding things. We're going to be producing these new proteins. And what this is going to end up doing is these proteins are going to start to bind to the surface of these neutrophils and these natural killer cells. Eventually, the initial interaction will cause the neutrophils to actually slow down and begin tumbling. They're going to begin rolling over themselves. And then eventually they will stop rolling, flatten out, and then enter into the tissue through a process that's known as diapedesis. So diapedesis is going to bring cells that are not normally in the tissue into the tissue in response to this inflammation that's happening. And the first major cell types you're going to get there are going to be your neutrophils. I'm going to put little dots in here so you remember that they are granulocytes. Natural killer cells are not. So those cells can make it onto the scene in as little as 30 minutes after inflammation is complete. So they can be there quite quickly. Now, neutrophils, when they get to the tissue, it's pretty simple. They are warriors. Their job really is to just sort of kill and break stuff. They're going to come in here. They're going to eat a few. Remember, they have 3 different ways of killing some of these invaders. They can either phagocytose them. They can't phagocytose nearly as many bacteria, for example, as a macrophage can. They're not the big eaters. Those are the macrophages. They can also secrete, so they can degranulate and secrete those granules of cytotoxic compounds, so they can destroy larger organisms that way. Don't forget, some of these cytotoxic compounds can also damage your tissue, and they can also secrete those nets and do that netosis type thing. So that's supposed to be a net. Sorry, my drawing isn't great. I'm not an artist. I'm a biology professor. So they have lots of different ways of killing things. So they're going to be super helpful. And there's 20 billion of them in your bloodstream, and you're going to end up with millions of them in the tissue very, very rapidly. And then once they kill a few things, they die and they become pus. They will also, when they offload that payload, they're going to begin secreting their own cytokines as well. This is going to help build the inflammatory response. Natural killer cells are, in this infection, early on, they might not all be all that helpful. Remember, natural killer cells are your innate immune system's antiviral weapons, right? So they are antivirals. And the way they do this is, in the case of viral infections, they're going to look for danger signs on the outside surface of your cells. And if there's enough of these sort of weird, wonky proteins or carbohydrates out there, they might tell that cell to kill itself. Now, natural killer cells can help in this particular bacterial infection in one particular way. Remember that natural killer cells and macrophages have sort of a weird interaction. So let's draw another macrophage down here. So here's another happy-go-lucky macrophage that has not been activated in response to this particular invasion just yet. But when this natural killer cell gets on scene, it can actually begin to secrete cytokines. And these cytokines can make this macrophage go from being happy-go-lucky, sort of garbage-collecting guy to now I'm activated. And in its activated state, it's going to begin to sort of follow these breadcrumbs, these cytokines, and begin moving towards the tissue. And eventually, it's going to become hyperactivated. And this hyperactivated macrophage is going to begin secreting a bunch of its own cytokines. And what's interesting is these cytokines can actually now interact with the natural killer cell. And this natural killer cell, for them, it's a signal to proliferate, which means to make more of themselves. So they begin to make more of themselves. And of course, those newly born natural killer cells begin to secrete their cytokines, and they begin to recruit even more happy-go-lucky macrophages. So what's going to happen here is this interaction between activated macrophages, hyperactivated macrophages, and natural killer cells helps to sort of build this immune response at a very early stage where we get more and more inflammation, the recruitment of more and more tissue-bound and blood-bound immune cells. And at this point, we have sort of this self-perpetuating process here where the innate immune system is really fighting hard to keep whatever this is in check, because remember, these bacteria that are in this particular case, they're not static. They're going about their business. They're fighting back. They're going to continue to divide, and this infection is only going to get worse as these guys begin to survive. We're going to need more and more cells. Now, if this is a full-blown infection that your innate immune system can't really control, we're going to need to get specialized help into the tissue. And that's going to come in the form of the adaptive immune cells. Now, remember, there's one cell here at the battle scene that can really help with this, and that's our friend the dendritic cell. Because the dendritic cell has spent its entire time collecting as much of this battlefield antigen as it can, and by now, it's pretty full up. So remember, it's going to hang out in the tissues for 6 hours or so and just collect as much of this stuff as possible. And at that point, it's going to go from the battle scene. It's actually going to hop into the nearest lymphatic vessel. And it's going to travel to the nearest lymphoid organ. So we'll say for our purposes that somewhere down here is going to be a lymph node where it's going to travel. And in the process, what it's going to do is it's going to begin getting itself ready for the process of antigen presentation. So this right here is strictly the innate immune response that would occur very early on in the first minute to 24 to 48 hours of an infection. And what's going to have to happen is we're going to have to start getting the adaptive immune system online. So what I'm going to do is I'm going to erase this particular scene right here. We're going to come back to it, but I'm going to erase this because what we need to start talking about is what's going to happen next when we get to the lymph node. So I'll erase all of this stuff. We will eventually get back to the battle scene here. And sorry, I see my background there. For those keeping track at home, yes, I am a tremendous Liverpool fan. While I erase this, and we'll get to our next picture, which is the lymph node. So let's get to the lymph node real quick, and let's draw that out. So remember, you've got this lymphatic vessel that's coming in, and it's going to lead into the larger lymph node. And in the center of that lymph node, well, you've got two parts, right? So we've got the outer part of the lymph node, which is referred to as the cortex. And this is where the B cells live. And we've got the paracortex on the inside, which is where the T cells live. Okay, so let's talk about what T cells are. So first, let's get our dendritic cell here. So here comes our dendritic cell. It's going to arrive here in the T cell zone. And it's going to be, at this point, I really love the term, and I love the term bedazzled. So it has taken, in that journey, it's taken all of that battlefield antigen, it's chopped it up into small peptides, and it's now presenting them in a particular cell receptor, cell surface receptor, that is called an MHC2 receptor. Now remember, there are 3 different types of MHCs. We really only learned about 2. The third one isn't really relevant to this conversation. MHC2s are only present on our 3 antigen presenting cells: our dendritic cells, our macrophages, and our B cells. The other type of MHC that's present on this dendritic cell is an MHC1 receptor. But the reason why we're focusing mainly on the MHC2 receptor is this is a bacterial infection. And if we looked at what was in the MHC1 receptor of this particular dendritic cell, we would find that it's only self-antigen. In other words, it's not interesting to most of these T cells that we're going to talk about in just a second. We'll get back to that in a minute. So let's talk about 2 different types of T cells. We have our helper T cells, and we have our killer T cells. And there's a couple ways of writing this. A very common abbreviation is CTL, stands for cytotoxic T lymphocyte. Sometimes people write TC, a cytotoxic T cell. However you want to abbreviate it, if you want to write killer T cell, I don't care, it's up to you. This is your drawing. So remember that these T cells all have their own unique, wonderful B cell receptor, or T cell receptors, right, on the surface. And this T cell receptor is what grants them that specificity. Remember, the 2 key characteristics of the adaptive immune system are specificity and memory. We'll get to the memory part in just a minute. So this T cell, there's going to be lots of helper T cells running around in here. Thousands of helper T cells are going to be running around in this paracortex of this lymph node. And this this dendritic cell is going to begin to trying to present antigen to all of these different helper T cells in hopes that it finds T cells that have a T cell receptor that will recognize its cognate antigen. So let's say, for instance, it has found this one. This is a helper T cell whose T cell receptor recognizes some of the antigen that this dendritic cell is presenting in its MHC2 receptor. So remember, there are a few steps needed to activate a naive helper T cell. Okay, so let's talk about what those are. The first thing first, the first step is, does that dendritic cell have the right information? So this is going to be conferred by the TCR. You need that T cell receptor to recognize its antigen, right? That's the first step. If this helper T cell here can't recognize its cognate antigen in that MHC2 receptor of that dendritic cell, then it's not a helper T cell that's going to aid in this infection. If this person has a bacterial infection and a helper T cell that recognizes Ebola proteins, we don't want that activated, right? So that's the first step. The second step is confirming that the information is coming from the right source. So the first step is it the right information? The second question is it the right source? And that is actually going to be conferred by another receptor on the surface of that helper T cell. It's the CD4 receptor. So remember, there's a CD3 co-receptor here, and then a CD4 receptor that goes out and actually talks to the MHC2 co-receptor on the surface of that dendritic cell. So we need the CD4 to recognize an MHC2. Why is it so important? Well, remember, helper T cells are only supposed to be activated by an antigen-presenting cell. Well, only antigen-presenting cells are allowed to have MHC2 receptors. So this step basically says, yep, I've got the right information, but not only do I have the right information, I have it from the correct source. Now, the third step in this process is a question of abundance. Is this an antigen that is present in abundance? In other words, is the thing that this helper T cell is going to be able to respond to, is there enough of it around that warrants this particular helper T cell and the B cells it might later on activate? Is there enough of it around to warrant this process? Now, this is going to lead to something called an immunologic synapse. So an immunologic synapse formation. This basically means there are going to be a lot of This basically means lots and lots of TCRs are being bound by their cognate antigen. It sends a very strong signal to that cell that yes, this particular helper T cell, or this T cell in general, is very, very important for activating, for helping to fight off this infection. The fourth step is a process that is called co-stimulation. So remember, co-stimulation is going to happen because a particular protein is going to be produced. It's called the B7 protein, is being produced by the dendritic cell, and that's going to interact with something called the CD28 receptor on the surface of the helper T cell. And this co-stimulation is sort of like a microphone. It helps to relay the signal and sort of supercharge that message that this particular helper T cell is needed for to help fight off this infection. Okay, so now that we have this helper T cell activated, this is the process of clonal selection. And the next step in this process is a question of clonal expansion. This helper T cell is going to begin making more of itself. And helper T cells are able to make several different types. So remember, a helper T cell that has been activated can give rise to more helper T cells. Okay, so it can make more helper T cells, and these are going to be needed both for this particular infection, but remember, about 10% of them will linger around in the tissues in many cases to form sort of long-term memory cells as well. So helper T cells. Now remember, there are different several classes. So there's Th1s, and there are Th2s. There are Th17s. So these will be useful for helping in the process of this infection. There is also the ability to form some of those memory central Ts. So memory Th cell. So these are CD4 positive memory cells. These guys will sort of hang out in the lymph nodes, and they're going to begin waiting for the next infection to come on. Remember that some of these helper T cells here will actually become memory T cells. That 10%, those are your tissue resident memory T cells that'll hang around in there. And then the final thing helper T cells can become later in the infection is a regulatory T cell. And these guys are the ones that help to put the brakes on an infection. Or an immune response. An important thing to remember is helper T cells never differentiate into a killer T cell. That's not the process. That process actually occurs in the thymus, where the maturation process occurs. And if you're doing your own drawing, you may want to put that on there. I don't put it on mine, but that's up to you. The other thing to think about is, where are these helper T cells going? So helper T cells can go to a few different places. The first one is going to go to the tissue. So these helper T cells here can go to the tissue to help fight the infection. Remember, that's we'll talk about that in a little bit, but hold that in the back of your head. So some of them are going to the tissue to help fight the current infection. The other place some of them will go is called the tour de lymph. That's my own term. I don't think anybody's ever called it that. The tour de lymph. That means they're going to circulate throughout the body, go to other lymph nodes where they might be needed to help get the adaptive immune system online, particularly by helping to activate naive B cells. And the third place they are going to go is to the T cell, B cell zone border. Okay? And we'll talk about why that's important. So some of these guys are going to begin heading over here. They're going to chemotext. Whenever they change what receptors they produce, they become suddenly sensitive to certain chemokines that draw them out to this particular portion of the lymph node. Now, the other thing I want to talk about is what if we change this process and we say that we're going to activate a naive killer T cell instead. Most of this stays the same, except really here it's not going to be CD4 and an MHC2. It's going to be a CD8 recognizing an MHC1 receptor, because remember, killer T cells are interested in what's in MHC1s. The other thing that we do know, I'm going to clarify that for you there. The other thing that we do know is that to get a full-blown killer T cell response that involves the formation of memory cells and so on and so forth, there's a fifth thing here. They seem to need some involvement from helper T cells. We know that in the absence of helper T cells, killer T cells can come online. They can begin fighting infections, but they're much more effective and much better at forming a memory when helper T cells get involved. And there is still some work to do to figure out that exact mechanism by which helper T cells aid in this process. So that's it for the T cells. The other type of cell that we need to get activated as part of the adaptive immune system is going to be our B cells. So forgive me while I erase some of this stuff, but I need to make room for our notes on B cells. So B cells, of course, are lymphocytes. I'm going to change colors slightly to clarify this a little bit for you. But remember that B cells live in a different part of the lymph node. They live in the cortex, right? So they're out here, and specifically, they're actually out here in these things, these little areas that are known as lymphoid follicles, okay? So just remember, these are the follicles that I've drawn here in blue. And follicles really consist of two types of cells. The first one is this sort of starfish-shaped cell called a follicular dendritic cell. And then around that, you're going to have lots of B cells. So remember, it's sort of a lymphoid follicle is really follicular dendritic cells surrounded by a sea of naive B cells. And also remember that a B cell has its own unique, wonderful B cell receptor that gives it specificity. So let's sort of blow this drawing up a little bit so we can see it a little bit better. So here is our FDC, and here is a single B cell. So again, like we saw with helper T cells, it's not going to be a single solitary B cell. There's going to be thousands of these things interviewing with these follicular dendritic cells. Now remember that follicular dendritic cells are able to bind to pretty much any antigen they want, but that's because they have a special receptor on their surface that's actually able to bind to complement. So what they're waiting for is for a complement opsonized antigen to flow through the lymph, from the lymph into this tissue, so that they can bind to that complement and then dangle all of this complement opsonized antigen out into the lymphoid follicle, so that the B cells are able to begin interviewing them with their B cell receptors. And activating a naive B cell is analogous to what happens with the T cells, except there's a few different steps here. Apologies, I just moved that picture by accident. I'm still getting used to this program here. So let's activate some naive B cells. And remember that the first step is very analogous. It's a B cell receptor recognizing its cognate antigen, right? So we've got that step happening. Again, that's the question of, is it the right information? A B cell shouldn't be activated in response to antigen it doesn't recognize. We can skip that second step, though, of T cells that involves the use of CD receptors recognizing MHCs. And the main reason why is B cells are lonely. They really only have two friends. They hang out with either the follicular dendritic cell, which is pretty much the only cell that will provide it with antigen, and helper T cells. So there really isn't that need to validate per se that it's the right source. The other thing to realize is this: the dendritic cell bringing antigen to the lymph node to present in an MHC receptor is confirmation from the innate immune system that whatever it's presenting is a problem. Remember, these are materials that have been bound to PAMPs before, foreign molecules. This has already been accomplished. It's been accomplished by the fact that that complement has opsonized this antigen before it was delivered through the lymph to the lymph node. So we've already got that information. So the next step, again, is a question of abundance. And in this case, we refer to it as cross-linking. Cross-linking is lots and lots and lots of B cell receptors, all recognizing their cognate antigen at the same location in the plasma membrane of that activating B cell. Okay? And the final step is, in most cases, unless we're dealing with a super antigen, is going to be co-stimulation. Now, this co-stimulation requires a helper T cell that recognizes the same cognate antigen. So how does this happen? Well, this newly activated B cell is also going to change its architecture, and it's going to begin migrating out here to the border between the paracortex and the cortex. So in the end, what you're going to have is a bunch of B cells that have lined up on this edge and a bunch of T cells that have lined up on the border between the T cell and the B cell zone, and they're going to begin this process of trying to figure out whether or not they share the same cognate antigen. And this kind of looks, I'll draw it over here, kind of looks like this. You've got this T cell with its T cell receptor. You've got this B cell that does have its B cell receptor, but it's not involved right now. Instead, what it's going to do is it's going to take whatever antigen it pulled off the surface of that particular follicular dendritic cell, and it's going to begin presenting it in its MHC2 receptors. Remember, B cells are also able to produce MHC2s and present antigen. Now, if this particular T cell then recognizes that, yes, this is my cognate antigen on the surface, it's going to become happy, right? And it's also going to go ahead and make that B cell happy as well, because they're going to have an interaction. First off, the helper T cell is going to provide something called CD40L, and that is going to fit into a CD40 receptor on the surface of that B cell. That should probably be blue to make it less confusing. So a CD40 receptor. And this is the co-stimulation between the two. This rescues that B cell from undergoing apoptosis. It confirms the clonal selection process, and we'll lead in a second. We'll talk about the clonal expansion process. But, so that's an interaction in this direction. But B cells are also going to produce that same B7 protein that co-stimulated the helper T cell in the first place, that fits into that CD28 receptor. And that actually is going to turn this helper T cell into a follicular helper T cell. And he's going to kind of move into that lymphoid follicle, and he's going to begin interacting with the follicular dendritic cells and the B cells, helping to co-stimulate and re-stimulate newly born B cells as they begin to go through this process of clonal expansion. So, of course, we're going to get more of these B cells cloned because they're going to go through that process of expansion. B cells basically divided into two different groups. You've got the majority of these are going to become plasma Bs. Plasma B cells are the ones that are going to begin producing antibodies. And then the other type is going to be, now these can be both short-lived and long-lived. So remember, the short-lived ones are going to begin producing thousands of antibodies per minute and launching them into your body to help fight the infection. The long-lived ones are actually going to go to the bone marrow, and their job really isn't involved in this infection, but they're going to spend, they're basically a form of memory cell that's going to spend a good deal of time, for a very long time, secreting low levels of antibodies into your system to help sort of recognize or stave off any future infection. And remember, some of those antibodies, if they bind to something that gets flushed into the lymph, they can again bind to the surface of follicular dendritic cells and then activate the other type of cell, which is that memory B cells, so those central memory B cells, which can be activated in response to a reoccurrence of this infection, which could be a few weeks down the road, a few years down the road, a few decades down the road in some cases. Also, these memory B cells will help to replenish the population of long-lived plasma B cells as they begin to age, become non-productive, and eventually die. Okay, so the other thing to remember is this is the case for like 99% of all B cells. There is that other pathway where cells can, or B cells can be activated by super antigens that cross-link a ton of BCRs and sort of bypass the requirement for that co-stimulation step. Now, at this point, let's talk about what those plasma B cells are going to be doing. So they're going to begin producing antibodies. So antibodies are absolutely amazing. They're possibly our body's best weapon in the defense of our body. So let's talk broadly about what antibodies do. So antibodies, 5 major things that they're going to do. They can opsonize. So again, opsonizing is going to make it easier for phagocytes to eat things. It'll make it easier for natural killer cells to recognize whether it's a virally infected cell. It will help eventually. Antibody opsonized antigen can also bind to the surface of FDCs. So opsonization is a very good thing. It's able to do something called agglutination. So some antibodies are good at agglutinizing microbes. This is where they force them to clump together. It sort of prevents them from sticking around. We'll talk in just a second. IgA antibodies are great at this, which is they sort of clump up all the bacteria, which is great because then they can be sort of blown out of your nose in the form of snot, or they can be excreted from your body in the form of feces. Another thing they can do is something called neutralization. So if you think about a virus needing to take those protein spikes and binding to the surface of one of your cell receptors, well, these antibodies can bind to those protein spikes and they can prevent it from binding to the receptor, and thus basically neutralizing it and preventing the virus from causing an infection. They can fix complement. So fixing complement is the ability to recruit and activate the complement cascade. This can be very important, particularly early on in an infection where we don't have all of the pieces in place. So remember, IgM antibodies, the first antibodies any B cell makes, those can actually be activated that way. Those are really good at doing that. And the fifth thing is antitoxins. So antitoxins bind to those exotoxins that are produced by some bacteria like Corynebacterium diphtheriae, Vibrio cholerae, Clostridium tetani, Clostridium botulinum. They all are harmful because they produce toxins. Well, certain antibodies can recognize those and pull them out of circulation so they can't harm your body. Now remember, there are 4 classes of antibodies. So there are 4 classes of antibodies. You've got your, we'll go in alphabetical order, IgA. Those are the ones that coat your entire mucus, anything that's a mucous membrane as well as your digestive tract. They're also the ones that get secreted in breast milk. These guys are particularly good at agglutination, if you need to remember that. Binding things, pathogens up and preventing them from helping them be removed from your body. You've got your IgE antibodies. IgE antibodies are typically produced in response to the activity of type 2 helper T cells and type 17 helper T cells. They're very good at helping to fight off parasitic infections, fungi, worms, things like that. However, they are also able to bind to mast cells, which can lead mast cells to degranulate and cause things like they can cause anaphylactic shock, and they're also responsible for allergies, things like hay fever and food allergies. You've got your IgG antibodies. These are the ones that are mainly in the bloodstream. There are lots of different classes of these that are specialized for either being very good at fixing complement or being very good at opsonizing and so on and so forth. They're the ones that are mainly found in the blood. They're also the longest lived ones, and they're the ones that are delivered via the placenta to fetuses and babies as they're developing in the womb. And the last one is the IgM. So the IgM is the first antibody your body makes. It's also really good at fixing complement. And it's also, as far as we know, evolutionarily the first antibody that vertebrates acquired or developed through the evolutionary process. So that's what's going on with your adaptive immune system. But we've got to circle back. We've got to get back to the scene of the battle where we have some stuff's going on, right? So we still got all these bacteria out here. I guess I won't color this in all the way and make you watch me do it. But we've got all these bacteria out here. And, we still have complement going to work. And we still have these macrophages that are doing their own thing. And we've got all these cells out here that are still doing stuff, right? Dendritic cells are still, sorry, I'm getting a little bit sloppy, are still collecting antigen. You do have your neutrophils out here, and you do have your natural killer cells out here doing their jobs. But what's going to start happening is new cells are going to begin arriving from the blood vessels, and these are the ones that are going to really help us with this particular infection. These include helper T cells. So helper T cells have the ability to secrete cytokines that can help, sort of direct what's going on with the infection. Big thing to realize is that helper T cell is going to need to be re-stimulated when it gets to the tissue. So how's this going to happen? Well, it's going to have an interaction with an activated, not a hyperactivated, but just an activated macrophage, because they're very good at presenting antigen, because they can also produce MHC2 receptors. And if this T cell is able to interact with some antigen on the surface of that macrophage, it will be re-stimulated, allowing it to go to work. If it's not re-stimulated, it'll eventually undergo the process of apoptosis and remove itself from this particular tissue because it's not needed to fight this infection. Upon receiving that signal, it begins to secrete a bunch more cytokines, which can help recruit and continue the inflammatory response and basically keep things going for as long as needed. We're also going to get some if this were a viral infection, we'd expect killer T cells to arrive. This is not a viral infection in this case. But what this killer T cell will be looking to do is using its TCR to interact with the MHC1 receptors of virally infected cells and cause those cells to undergo apoptosis should they have been virally infected. Eventually, we would also start getting antibodies. So these are being secreted. So these antibodies, I'm not drawing them to scale, obviously. Antibodies are just proteins. Antibodies will begin secreting into the tissue. Those will begin binding to the surface of these bacteria, and this would begin to further aid in the infection. So, like I said, this is just a sort of one way to learn the immune system. I find my students have been quite successful in doing it. Use whatever colors you want. Add different layers to it. You're welcome to do this drawing however you want. But one of the things that I think it helps my students be able to do, and it's helped them in the past, is you start to put pictures with it. You start to put colors to it. And the more of your senses that you engage in the process of doing this, the better off you'll be. I've actually had students go so far as to use scented markers to do this, so that their blue innate immune cells always smell like blueberries, and their green helper T cells and killer T cells always smell like lime, and their antibodies always smell like raspberry or something like that. Things you can do to engage all of your senses. I hope this helps. I'm sorry that my drawing isn't great. I'm not the world's best artist. Plus, I'm not used to drawing on a tablet like I am to do this. But again, I hope it helps. Please feel free to add more information to this. Please feel free to, but I would say this is sort of the minimum distance you want to go if you're going to talk about all the things that we discussed in my videos. Thank you so much for taking a look at this. I hope it does help. If you have any questions, feel free to drop some comments in there, and I can give you some advice on that. If you're one of my students, you're also welcome to shoot me an email, and I'm willing to help. Thank you guys again so much for tuning in as we try to learn the immune system. I will talk to you very soon. Bye.